Hello everyone, I'm Dr. Gauri Prabhu. I am a junior resident in the Department of Pathology. So today's topic is apoptosis. The objectives of today's lecture is, by the end we are supposed to know the definition, the purpose of why apoptosis happens or how it happens, and the morphological changes that are seen, and let's discuss some of the important questions by the end. So what is apoptosis? It is a type of cell death that is induced by tightly regulated suicide program in which the cells destined to die, they activate the intrinsic enzymes that degrade the cell's genomic DNA and nuclear and cytoplasmic proteins. So this is the definition you must learn. But what exactly is apoptosis? So it's a programmed cell death. Okay. So how does the cell death occur? It occurs by activating the intrinsic enzyme, and that enzyme will degrade all the DNA, the cytoplasmic and nuclear proteins, and that's how cell death occurs. The causes of apoptosis, there are two situations apoptosis can occur. One is physiological and pathological. So apoptosis in physiological situations it serves to eliminate the cells that are no longer needed for our body. Okay, it's seen during development. Like if there is any error in apoptosis, it may lead to birth defects. So here apoptosis acts as a sculptor of human body or the fetus. Okay, and involution of hormone dependent tissues in withdrawal of hormone. For example, lactating breast after weaning, what happens? And next is elimination of potentially harmful self-reactive lymphocytes to prevent immune reaction against one's own tissue. Next is apoptosis in pathological situations. Here apoptosis eliminates the cells that are injured beyond repair. Okay, so if a person is getting exposed to radiations repeatedly, or if he is taking cytotoxic anti-cancer drugs or repeatedly under hypoxic conditions, then there will be DNA damage, and if that DNA damage is unrepairable or beyond repair, then what happens? Our body says it's difficult to repair this DNA, we will have to kill this cell. So it undergoes apoptosis. If our body doesn't do that, what happens? The DNA will undergo mutation, and later on the effects will be much worse. Okay, so next is accumulation of misfolded proteins. So when does misfolded proteins occur? When there is any mutation in the gene, or due to certain external factors, if the proteins are misfolded, and more of such misfolded proteins, they get accumulated in the endoplasmic reticulum, there will be endoplasmic reticulum stress, and in such cases that cell which has ER stress will undergo apoptosis, and this is seen more and more commonly in degenerative diseases of a central nervous system. And then it's also seen during certain infections. During viral infections we see apoptosis. Here it can be due to the host's immune response itself, or it could be due to the virus that may induce apoptosis. Okay, so mechanisms of apoptosis, there are two pathways through which apoptosis occurs. One is extrinsic pathway and the other is intrinsic pathway. So the extrinsic pathway, which is also called as death receptor initiated pathway. So this pathway is initiated by the engagement of plasma membrane death receptors on a variety of cells. Okay, so the death receptors, they are the members of TNF receptor family, and they contain cytoplasmic domain, which is also known as the death domain. Okay, and it is essential, this death domain is essential for delivering apoptotic signals. So some of the examples are TNF receptor family members, like type 1 TNF receptor, or FAS, that is CD95. So this FAS, it is a death receptor that is expressed on many cell types, and the ligand for FAS is the Fas ligand, FASL, that is Fas ligand. This Fas ligand is expressed on T cells. It's expressed on T cells that recognize the self antigens, and on some cytotoxic T lymphocytes which kill the virus infected cells and tumor cells. Okay, so when this Fas ligand binds to Fas, 3 or more molecules of Fas, they are brought together. So 3 or more molecules of Fas, they are brought together. When Fas ligand binds to Fas, 3 or more molecules of Fas, they are brought together, and their cytoplasmic death domain. So this is the cytoplasmic death domain. These cytoplasmic death domains are also brought together, and they will provide a site for the binding of adapter protein, which is known as Fas associated death domain. Okay, Fas associated death domain. So Fas associated death domain that is attached to the death receptor, in turn it binds an inactive form of caspase, that is caspase 8. It binds inactive form of caspase, so it is called as procaspase 8. This is inactive. Later on it undergoes cleaving and it becomes active caspase 8. It will undergo cleaving and it will become active caspase 8, and then further on it will activate other caspases, executioner caspases, and which it will lead to apoptosis. So as we have already studied, the Fas ligand is expressed on the T cells that recognize the self antigens. The Fas ligand will then bind to Fas death receptor that are present on the target cells. So cytoplasmic death domains of Fas, they provide a binding site for an adapter protein called as Fas associated death domain, or FADD. It will then activate caspases and endonucleases. Okay, so the caspase that is activated here first is caspase 8. So that will cause protein breakdown, and endonuclease, it will lead to DNA fragmentation, and all of this leading to apoptosis. Okay, so this is the extrinsic path. So now moving on to intrinsic pathway. So when the cells are deprived of survival signals, or if their DNA is damaged beyond repair, or if there are any misfolded proteins in the endoplasmic reticulum, then what happens? There will be P53 activation, and the BH3 only proteins, okay, this BH3 only proteins, which are the sensors that are present on the mitochondrial membrane, they act as the sensors of cellular stress and damage. Which one? The BH3 only proteins. The BH3 only proteins, they act as the sensors of cellular stress. They will cause upregulation of pro-apoptotic proteins like BID, BAD, BAX, BIM and BAC. So these pro-apoptotic proteins will increase the mitochondrial membrane permeability, leading to leakage of cytochrome C from the mitochondrial inner membrane into the cytosol. Okay. Then the cytochrome C that is leaked into the cytosol will bind with APAF, that is apoptosis activating factor, that is known as apoptosome. It is known as apoptosome. Okay, that is known as apoptosome. So that will activate the caspase. That will activate the caspase 9. Okay, it will activate the caspase 9, and this caspase 9 will activate other caspase cascade, and finally it will activate the executioner caspase, that is caspase 3, and there will be activation of endonuclease as well. So here the caspase involved is caspase 9, and finally the executioner caspase is caspase 3, and then endonuclease is activated. This will lead to protein breakdown and DNA fragmentation, which will lead to apoptosis. So this is the intrinsic pathway of apoptosis. All right. So the morphological features of apoptosis, first of all, what you see is cellular shrinkage. Then there will be cellular blebs and apoptotic bodies, chromatin condensation. On HND, there will of the apoptotic cells, they appear as round or oval mass of intensely eosinophilic cytoplasm with fragments of dense nuclear chromatin. So just remember, any apoptotic cell will have hyper-eosinophilic cytoplasm and there will be fragments of dense nuclear chromatin. Okay. So here this is a picture, HNA picture, which is showing hyper-eosinophilia. Alright, and this is an electron micrography. Here we can see that this is the nucleus. You can see there is nuclear condensation and all this condensed nucleus has come and it's sitting under the nuclear membrane. These are all the fragmented nucleus. Okay, this is the phase contrast micrography. In this you can see these are all the cytoplasmic blebs. Okay, so here look at this, these are all the fragmented parts of the nucleus. Okay, so this is about the morphological changes. So one thing is you have to tell about hyper-eosinophilia. There will be cellular shrinkage and hyper-eosinophilia of the cytoplasm. There will be nuclear condensation, nuclear fragmentation, cytoplasmic blebbing. Okay, so one MCQ I have posted here. So this is concerned with the intrinsic pathway of apoptosis. Which is concerned? Is it cytochrome A, B, C or D? We all know that it is cytochrome C. So cytochrome C is present on the inner membrane of the mitochondria, which will leak out into the cytosol of the cell when pro-apoptotic proteins will get activated. Okay, so this will then bind to APAS. It will then bind to APAS and form apoptosome and will activate the caspases and then the endonucleases. This is the right answer. So you should know the other questions that may be asked here is it could be describe apoptosis for 10 marks, or they may ask just the mechanisms of apoptosis or the examples of apoptosis. So we have to describe all of these. Yeah. All of these information was taken from Robbins and Cotrans. Thank you.